discussing more details how glycolytic pathway is controlled. So here, what you are seeing is that glycolytic pathway is tightly controlled. And what is the process by which it can be controlled is that there are three reactions which are irreversible reaction. One is a hexokinase, another is a phosphofructokinase, and third one is a pyruvate kinase. You look at all these reactions, they are all, either requiring an ATP or they are producing ATP. So they are the one which are energy requiring or energy generating steps. And let me tell you that these are the ones which are being controlled in most of the metabolic reaction. That's why we are taking glycolysis as one example and trying to show you how the metabolic pathway can be regulated or controlled. So here you will see that the phosphofructokinase is the main enzyme which is controlling the reaction. So this phosphofructokinase, and what is the role of phosphofructokinase? It is the enzyme which converts fructose 6-phosphate to fructose 1,6-bisphosphate. And as we said that there could be control by different mechanism, today we are going to talk about how phosphofructokinase is being controlled. So yesterday we have talked a little bit about this. So let us come to the controlling molecule. The molecule which controls the enzyme is fructose 2,6-bisphosphate. Now this fructose 2,6-bisphosphate is different than fructose 1,6-bisphosphate. Why? Because fructose 1,6-bisphosphate, when it is formed, it will go, in the forward direction of the glycolytic cycle or glycolytic pathway, where it is going to produce two molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. But this molecule is not for energy generation. It is not for, going further for metabolism in glycolysis, but rather this molecule is for controlling the glycolysis. And let us see how it works. So actually, this is acting as an allosteric activator. So when I say allosteric activator, what do you understand? That means this will go bind with the enzyme, and once it binds with the enzyme, it will either activate or deactivate. Here, what it does is that it is going to convert the enzyme from tense state to relaxed state. And when it is a relaxed state, that will be active. When it is a tense state, it is inactive. So formation and presence of this fructose 2,6-bisphosphate is the one which is a controlling. And let's see how it controls. Of course, I have given you the kinetics, how it works, but let us talk about how this works. So the regulation is done by an enzyme, which is a bifunctional enzyme. And what it does, it synthesizes and degrades fructose 2,6-bisphosphate. So the level of fructose 2,6-bisphosphate itself is controlled by an enzyme, and this enzyme is a bifunctional enzyme. So what will be happening? So there are two enzymes. One enzyme is the which phosphorylate fructose 6-phosphate to fructose 2,6-bisphosphate, and another enzyme which will convert this fructose 2,6-bisphosphate to, so one enzyme will convert this into this molecule, and another enzyme will convert this molecule into this molecule. So actually, what will happen that the level of fructose 2,6-bisphosphate can be controlled in the cell. And how it happens? It happens because of the two enzyme. The enzyme which converts, which is responsible for formation of fructose 2,6-bisphosphate is phosphofructokinase 2. So what phosphofructokinase 2 does is it makes the fructose 2,6-bisphosphate. How it does? You have fructose 1-phosphate, and then if its concentration is high, what it will do is that it will make fructose 2,6-bisphosphate. I'll show you in the diagram how it works. On the other hand, fructose 2,6-bisphosphatase, what it will do is that whenever this molecule, see, look at this, this is a substrate. Okay, so this substrate is getting hydrolyzed to fructose 6-phosphate. That is what I said. This substrate can be hydrolyzed to fructose 6-phosphate. And what works here? The enzyme is phosphatase. I'll just show you in the diagram. The major interesting thing is that the one which forms fructose 2,6-bisphosphate or which degrades fructose 2,6-phosphate, both are present in the same complex. So you have a an enzyme which is a mixture of or is a combination of fructose, uh, this phosphofructokinase 2 and fructose bisphosphatase 2. So the one which controls synthesis or controls degradation is present in same polypeptide, same enzyme. So say that is the one which is having both the function. And that is why it is called as a bifunctional enzyme. So bifunctional enzyme will have a kinase and a phosphatase. So kinase will synthesize 2,6-bisphosphate and phosphatase will degrade into from 2,6-bisphosphate to fructose 1-phosphate. So that's the way the mechanism happens. So this bifunctional enzyme is in isomeric form. Isomeric form or isoforms are the enzymes which differ in the size and kinetics, as well as they are, have a different immunological properties. So they differ in terms of their immunological properties as well as regulatory property. In liver, so you may find, so isoenzyme will be the same enzyme catalyzing the similar reaction, but what will happen, they will have difference in their kinetics, they may have difference in their regulatory properties. So one may be regulated by one mechanism, another may be regulated by another mechanism. So this overall reaction is very complex. So first let us see how it works. So here I'm showing you that this is a membrane. This is the membrane where you can have. So here, whenever glucose concentration is low, so this is the one which is going to control the overall reaction, and this synthesis and degradation of fructose 2,6-bisphosphate is being controlled. So how it controls? Actually, the signal comes from a hormone, which is a glucagon. What it does is that it binds with a glucagon receptor on the membrane. Then it will activate a protein, which is known as a G protein. And don't worry, though it is a complex, but we will be referring and discussing this when we talk about the hormones and signal transduction. That time we'll again discuss the same mechanism. So you have hormone which binds with the membrane surface, which is a receptor. This receptor will activate a protein which is known as a G protein. And once G protein is activated, it activates adenylate cyclase. And what adenylate cyclase does is that it forms cyclic AMP from ATP. So you have ATP present in the cell, this will form cyclic AMP. This cyclic AMP is the one which is a secondary messenger. Now, so message has come from glucagon, which is a primary messenger or a hormone. It will activate cyclic AMP, which is a secondary messenger, and this secondary messenger will do the job. So what is the job? It will activate protein kinase A. A is because it is AMP which is activating. It could be C also, where in a different mechanism where calcium activate, we call it the protein kinase C. So don't worry, right now we are just discussing about protein kinase A. So when protein kinase A is activated, what it will do is that it will take care of this complex. So you have a complex which has a both activity. One is a phosphofructokinase 2 activity, and another one is a this phosphatase activity, which removes phosphate group from here. So once you have this, what it will happen? It will phosphorylate. ATP will give its phosphoryl group, convert into ADP, and the complex is getting phosphorylated. Now, what happens that you have seen that this is everything is happening in the low concentration of glucose. So when glucose concentration is low in blood, what it should do? It should, in fact, stop the further metabolism of glucose. Rather, it should, capture the glucose for much more important activity. So what happens that at this time, this fructose 2,6-bisphosphate will be dephosphorylated, and when it is dephosphorylated, you get fructose 6-phosphate. That is what I told you earlier as well. So during this formation, what happens? Now this cannot stimulate phosphofructokinase, because what can activate phosphofructokinase? This molecule can activate phosphofructokinase. So once it is converted into fructose 6-phosphate, it will not activate phosphofructokinase. And once it does not activate phosphofructokinase, what will happen? The glycolysis will stop. On the other hand, after a certain period of time, when glucose concentration comes back to normal in the cell, glucagon will not be secreted from the as a hormone. So this pathway is not happening. Once it is not happening, what will happen? Slowly, you have the same enzyme, you have another enzyme. What it will do is that uh, they will phosphorylate it, and after phosphorylation, what you get is your fructose 6 phosphate. So this fructose 6 phosphate, which has been formed here, this fructose 6 phosphate, what it can do is that it can stimulate this process. And what it will do is that it will phosphorylate, and then after phosphorylation, what it activates is it activates PFK2. And PFK2, that is phosphofructokinase 2, what it will do is that it converts fructose 6 phosphate into fructose 2,6 bisphosphate. And what it will do at the end, this is the one which will stimulate phosphofructokinase, and when phosphofructokinase is activated, it will increase the pace of the reaction of glycolysis. Does it seem too complex to you? Or it is understandable? Now, what I'm trying to say here is that you have one complex which can either activate or deactivate fructose 2,6 bisphosphate, phosphatase. So what you have is that you have formation or degradation of fructose 2,6 bisphosphate, which is regulated by presence of glucose. Means whether glucose is a high concentration or low concentration. If it is being regulated, unless it is present, if it is degraded, then it will not activate this enzyme, phosphofructokinase. If it is present, it will activate phosphofructokinase enzyme. So it's very simple. If glucose is more, that means glycolysis should go on. If glucose is less, glucose should be conserved. So you must be wondering what is conservation means. Conservation means that if glucose is less in our system, it should go to the brain, provide energy to the brain, rather than doing metabolism for other purposes. So glucose concentration is tightly regulated by this bifunctional enzyme. Now tell me if you have not understood anything. Just to give you a written concept, here it is given very clearly that when fructose 6 phosphate leads to the higher concentration of fructose 2,6 bisphosphate, which in turn stimulates phosphofructokinase. This process is called as a feed-forward stimulation. That means when you have high concentration of fructose, it will direct phosphofructokinase to work more so that you can get fructose 1,6 bisphosphate. Now, how it does is that it does by increasing the high concentration of fructose 2,6 bisphosphate, which is a allosteric regulator or stimulator. The activities of phosphofructokinase 2 and fructose bisphosphatase, they are reciprocally controlled by phosphorylation of single serine residue. So that is what I have shown here. This is controlled by a single enzyme. So this I have shown you two to show you that it has got activity of phosphofructokinase 2 as well as fructose bisphosphatase. So this is controlled by a single enzyme. So there is a single enzyme. Both of them, they are present. You may be wondering how it is done. It is the fusion of two genes which is leading to the synthesis of these two enzymes. So on one hand, excess of glucose can activate the formation of this fructose 2,6 bisphosphate, or alternatively it can lower the formation. So actually the formation of this molecule fructose 2,6 bisphosphate is the one which is controlling the reaction for the formation of fructose 1,6 bisphosphate. There are, one has to very clearly understand the difference between fructose 1,6 bisphosphate and fructose 2,6 bisphosphate. 2,6 bisphosphate is the one which is regulating the enzyme, and 1,6 bisphosphate is the one which is actual product of the reaction of phosphofructokinase 1. Okay, now tell me, have you understood?
Yes, sir, somewhat.
Somewhat. Okay. It is, it is difficult to understand. I understand why it is problem because of the similar nomenclature. But think in this way. Very simple thing which you can think about is that when glucose concentration is high, it should be metabolized, and when it has to be metabolized, you should have high concentration of fructose 2,6 bisphosphate. Once it is there, then only it will activate phosphofructokinase, and once it activates phosphofructokinase, then phosphofructokinase will form fructose 1,6 bisphosphate, and that will go for further metabolism. That part is clear. On the other hand, when glucose concentration is low, the whole complex is, is inhibited. And if it is inhibited, your phosphofructokinase, whatever is already present in the system, that will be degraded. That will be forming fructose 6 phosphate. And once fructose 6 phosphate is present, they will not be activating the enzyme for further glycolysis. So it's as simple as that. It's not very complex, but it requires you to read and understand. Now, as I said, that this enzyme is the one which is a pacemaker enzyme. That means it is also going to affect other enzymes. So when phosphofructokinase is inactive, suppose I block this. Let me just go back to the reaction to show you the exact what happens. Yeah, here I'm referring to a very crucial reaction so far, whatever we have talked about, is the reaction, this one, this step, where I'm talking about how fructose 6 phosphate is converted into fructose 1,6 bisphosphate. Now, if I say that if I have fructose 2,6 uh, phosphate, it is 2,6 phosphate, that is the one which will control this, it will go ahead. If it is not there, what will happen? It will be converted into this by enzyme, and once it is there, probably they will not activate the reaction. So what is important here is that fructose 6 phosphate synthesizes fructose 2,6 bisphosphate. Once fructose 2,6 bisphosphate is there, this will activate. So what is happening? It is a feed-forward reaction where it is activating the enzyme phosphofructokinase for synthesis of fructose 1,6 bisphosphate. Now what I'm saying is that, so we have discussed about this mechanism. Now what I'm saying is that if I block this step here, what will happen? Fructose 6 phosphate will be accumulated, and if it is accumulated, it will inhibit this particular reaction. So, and that reaction is catalyzed by hexokinase. So let us come back to that reaction. So when phosphofructokinase is inactive or inactivated because of the lack of fructose 2,6 bisphosphate or so the concentration of fructose 6 phosphate rises. In turn, the level of glucose 6 phosphate rises because equilibrium with fructose 6 phosphate, because that two reactions are in equilibrium. Hence, the inhibition of phosphofructokinase leads to the inhibition of hexokinase. Is that clear? So when
Yes,
I
sir.
block the enzyme, which is in the forward reaction, what will happen? If the substrate accumulates, what will happen? It will also block the reaction which is at the starting point. And now what happens at this point of time, there is another very interesting thing happens, that in liver, uh, just, okay. So what is happening here is when you are blocking this phosphofructokinase, or it becomes inactive because of the lack of your activator molecule, which is nothing but fructose 2,6 bisphosphate. Under those conditions, hexokinase becomes inactive because of the high concentration of your this enzyme fructose 6 phosphate, and because of that glucose 6 phosphate also rises. So actually what you are looking at is the product of the reaction is inhibiting the enzyme. You know about this, that most of the time product of the reaction inhibits the catalyst. So however, in the liver, in keeping its role as the monitor of blood glucose level, possesses a special enzyme of hexokinase. So a specialized isozyme of hexokinase is known as a glucokinase. So if I am inhibiting hexokinase, what should happen? When I'm inhibiting this, what is done is that glucokinase is another enzyme which is present in the cell, and it is not the one which is inhibited by glucose 6-phosphate. So even if I inhibit hexokinase, this enzyme, even if I inhibit, glucokinase is present. The role of glucokinase is to provide glucose 6-phosphate for the synthesis of a glycogen. What it means, or for storage, which is finally is going to be stored as a carbohydrate material. This part we have talked about. So what happens that when you are inhibiting hexokinase, which inhibition is coming because of the excess of fructose 6-phosphate present in the system, this kind of a mechanism can be taken care of where glucose 6-phosphate can be produced for glycogen synthesis. So so that is where, though the enzyme is inhibited, there is another pathway which has been generated because of the isozyme. As I said earlier also, the isozyme is the one which will have different kinetic mechanism. So glucokinase will be the one which will be synthesizing glycogen molecule, and that will be stored. So if it is not metabolized, if your ATP concentration is very high, your glycogen synthesis will occur. So this is a very interesting point, that phosphofructokinase rather than hexokinase is a pacemaker of glycolysis. So that means hexokinase, though it is the first enzyme of the pathway, it is not the one which is deciding the pace of glycolytic reactions. What is deciding pace of glycolytic reaction is rather phosphofructokinase, because it is controlling even hexokinase. So that is where the phosphofructokinase is considered to be a pacemaker enzyme, or the one or enzyme which is deciding about the pace of the reaction. The first irreversible reaction unique to glycolytic pathway, the committed step, is phosphorylation of fructose 6-phosphate to, it is the same reiteration that phosphofructokinase is the one which will commit the glycolysis, and this commitment is occurring because of the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate. In general, the enzyme catalyzing the committed step is the metabolic sequence, which is the most important. That's what you have seen, that the conversion of fructose 6-phosphate to fructose 1,6-bisphosphate is a very important and committed step. Now there is another enzyme which is responsible for irreversible reaction, which is a pyruvate kinase. So what pyruvate kinase does is, if you recall, pyruvate kinase is the one which is responsible for the synthesis of pyruvate, but at the same time, it is also responsible for generation of ATP. It is the last step of the glycolytic steps, and what it does is that it is again being controlled. So now we are going to talk about pyruvate kinase. So pyruvate kinase is a tetramer, 50 kD subunit. Right now it is not very important, but it's important that it is encoded by different genes and are present in mammals in three different forms. One is L type, another is M type. These are, okay, so the one type, L type, and the third type is a mixed type, which I'm not talking right now. So L type refers to liver. So the one which is expressed in liver is different from the one which is expressed in the muscle and brain. Now these two, they will differ in terms of their regulation. Regulation means one will be regulated, another will not be by the same mechanism. So ATP allosterically inhibits L and M form of pyruvate kinase to slow the glycolysis when the energy charge is high. So actually this reaction is the one which is synthesizing ATP and also being controlled by ATP. So what will happen if you have excess of ATP? It will allosterically inhibit the pyruvate kinase. So, so what will happen that this is the step which is responsible for the pyruvate synthesis. So what happens that when blood glucose level is low, glucagon triggered cyclic AMP cascade leads to phosphorylation of, you have seen this path, that glucagon triggers the cyclic AMP, and this is the one which also leads to the phosphorylation of pyruvate kinase. So you look at how the enzyme is being affected by the hormones. So that's the way it controls all the enzymatic reactions. So let us see the mechanism. It's very simple. I have given you in a reaction which shows that hexokinase and pyruvate kinase, they set the pace of glycolysis So first is the one which is the pyruvate, the phosphofructokinase, which is responsible. So major job is done by phosphofructokinase, but these two, hexokinase and pyruvate kinase, also help in the glycolysis. Once you have taken care of phosphofructokinase, these hexokinase and pyruvate kinase can also lead to the pace of glycolysis. So what it does, when there is a high blood glucose level, under those conditions, this reaction occurs. You have a phosphorylated pyruvate kinase, which is less active. It becomes active. So when how it becomes active? It's one phosphoryl group has been removed. You have enzyme which is called as a phosphatase. So phosphatase, it will remove phosphate from pyruvate kinase. Now, so suppose there is a phosphate which is associated with enzyme. This will be removed. So this has been removed. Once it has been removed, what you can see is that it becomes dephosphorylated and it becomes more active. So once it becomes more active, the phosphoenol pyruvate to ADP and proton, they will form pyruvate. So this reaction happens. Now what happens when the glucose level is low? This more active enzyme will become less active by enzyme kinase. And this kinase will phosphorylate. This phosphoryl group has been attached. It becomes less active and this reaction slows down. Now, what can activate this reaction? If I have high level of fructose 1,6-bisphosphate, that will activate this reaction. On the other hand, if I have high level of ATP and alanine, they will deactivate or lower this reaction. So you have seen that how the control of pyruvate kinase occurs, how the control of phosphofructokinase occurs. So this completes the information about how glycolytic pathway is being controlled. So is it clear to you?
Yes, sir.
Okay. So now, after discussing this, we will be starting about the third aspect of glycolysis. So let me just go back to the, okay. So here, what we have talked about is that pyruvate can form ethanol, or it is also possible that pyruvate can form lactate. And third option given was that pyruvate can form acetyl-CoA, and this acetyl-CoA is the one which will go for further reaction. So now I have discussed about how pyruvate is formed, how pyruvate is being controlled, and now I'm going to talk about how acetyl-CoA can be further metabolized. Further metabolism of acetyl-CoA occurs only when molecular oxygen is in excess, and that is held by the process which is called as a citric acid cycle. So acetyl-CoA is further metabolized by citric acid cycle. So we will talk about that. Okay. So now let us so now I'm sharing another slide which is about the citric acid cycle. Is it visible?
Yes, sir.
Yes, sir.
Okay. So now you have seen that the acetyl-CoA has formed from the your pyruvate. So we will first talk about how acetyl-CoA is formed from pyruvate, and then we will talk about how this is being metabolized. Have you done this in microbiology, in microbial biochemistry?
Yes, sir.
Complete citric acid cycle?
So not the complete part, but like in brief.
Okay, so I'll go quickly. How about glycolysis? That you must have done in microbiology also.
Yes, sir.
Okay, but do you go for the details of the control, etc.?
Not exactly, sir.
Okay, so here the what I'm trying to do is that I will quickly discuss about the citric acid cycle, and we will be focusing on how the flux of the carbon, they are moving from one pathway to another pathway. So I think this will be very easy for you because you have already understood this. So I'll go very quickly about the citric acid cycle because it's a very important component of the metabolism. So citric acid cycle here, acetyl CoA. So do you know where this acetyl CoA is being formed from pyruvate to acetyl CoA? Is it in cytoplasm or somewhere else? Okay, I have written it. It is in mitochondrial matrix. So what happens? The pyruvate from glucose to pyruvate, this part is happening in cytosol. Now pyruvate through a special transporter enters inside the mitochondria, and in mitochondrial matrix this reaction occurs. And this is a very complex reaction. I will not go in very details of this reaction, but here there are three enzymes which are catalyzing the same reaction. And you will learn that these enzymes, they are complex enzyme, they are present in one unit, and that's the way the reaction proceeds. So what happens that the pyruvate is converted into acetyl CoA. What is important here is that the coenzyme A and NAD plus. What is NAD plus? You have just seen it in glycolysis. What is this NAD plus?
So it's an activated electron carrier.
Uh, okay, this is the one which is coming from your nicotina, say niacin, which is a vitamin. So it is a coenzyme. So this is required for the enzyme to function. So here what you have is that you have a pyruvate, you have a coenzyme A. Coenzyme A is again a form of vitamin, which is is a coenzymic form, and these two are involved in synthesis of acetyl CoA, carbon dioxide, and what will be the resultant is NAD plus is forming NADH. So if you look at this reaction, I'll just give you the complexity. These are the complex structure of thymine pyrophosphate, which is required. It is a lipoic acid. So the most important thing is that what happens when pyruvate is converted into acetyl CoA. Pyruvate is decarboxylated first, and then this molecule is transferred, and then what you have is that the coenzyme A gets attached with this acetyl group, and then you form acetyl CoA. Here, the kind of other coenzyme which is required is TPP, lipoic acid. So I will try to not go into detail of the complex reaction, but let us see that what are the three enzymes involved. So it is given for E. coli. You have E1. E1 is the pyruvate dehydrogenase component. So the same enzyme, which is known as the pyruvate dehydrogenase complex, will have three integrated enzymes. Enzyme 1 is a pyruvate dehydrogenase, enzyme 2 is dihydrolipoyl transacetylase, and enzyme 3 is dihydrolipoyl dehydrogenase. So there are three enzymes which are responsible for conversion of pyruvate to acetyl CoA. So the job of the first enzyme is being helped by thymine pyrophosphate. This is a coenzyme, and it will be carrying out oxidative decarboxylation of pyruvate. Why I'm calling it oxidative decarboxylation? Because it is involving both oxidation reaction as well as a decarboxylation reaction. So the oxidation reaction is the reaction where your pyruvate is converted into acyl group. And what else is happening here is decarboxylation. So from pyruvate, one carbon dioxide is being removed. So actually what you are learning here is that you are converting three carbon molecules. So acetyl CoA is three carbon molecule, which is converted into two carbon molecule. Sorry, acetyl CoA becomes two carbon molecule from pyruvate, which is a three carbon molecule. So this two carbon molecule enters in the citric acid cycle. And once you have transferred your acetyl group to coenzyme A, the next step is a regeneration of lipomide. So this FAD, which is responsible for catalyzing the reaction of dihydrolipoyl dehydrogenase, it regenerates the oxidized form of lipomide. So then it is again available for transacetylation reaction. So with the help of these three reactions, your pyruvate is converted into acetyl-CoA. There is steps I'm not going to detail. In fact, you don't require too much of complexity of the enzymatic reaction because I have already discussed for others. So now we'll move on. The only thing which you need to understand is that pyruvate is converted into acetyl-CoA in the mitochondrial matrix, and this is a conversion of a carb to 2-carbon unit. Now, it's a very important step which we have understood in the introduction to the metabolism. Acetyl-CoA is not only formed from pyruvate, it is also formed from, say, amino acids. It can also be formed from your fatty acid. So this is the one which is a central molecule, 2-carbon molecule, which is entering in the cycle, and it is the theme for the energy generation. Now, this reaction is giving you the complexity of the reaction. On one hand, a citric acid cycle is producing different molecules and produces the NADH, which could be source of electron and having high electron transfer potential, and that's why I can generate energy at the later stage. You do not see any reaction where ATP is being synthesized, except for the one which you will learn in the succinyl-CoA to succinic acid. Other than that, this cycle is also producing at different step different molecule. For example, citrate can produce fatty acid and sterol. Alpha ketoglutarate can produce glutamate. It is a transamination reaction. Your amino group is a transferred and keto group. So instead of keto group, if I attach amino group, it becomes glutamate. Once glutamate is synthesized, it can synthesize other amino acid and purines. What is a purines? What are purines? Here I have written pyrimidines as well, purines and pyrimidines. These are bases which are responsible for the synthesis of nucleic acids. So in nucleic acid, you have the purine and pyrimidine bases. So these purine and pyrimidine bases can also be synthesized from the intermediate of the TCA cycle. So on the other hand, we can say that TCA cycle is amphibolic pathway, a pathway which is not only is involved in catabolic reaction, it is also involved in anabolic reaction. So you see anabolic reaction, synthesis of fatty acid sterol. You see anabolic reaction where amino acids are synthesized as well as a nucleic acid is being synthesized. You see other molecule like porphyrin, heme, chlorophyll, they are being synthesized. You can also see that other amino acids. So there are different steps where they are being utilized. Now you can see a very interesting point that some of the intermediates of the citric acid cycle can leave the pathway for the synthesis. So now question is that if I have one molecule of citrate and if I remove it, will cycle continue? And if not, then how it should be continued? Answer is that if I remove any of the molecule from here in molar concentration, they will stop the reaction. So catalytic regeneration of oxaloacetate is very important because 2 molecule of acetyl-CoA, 2-carbon unit enters here, and you will see in the next steps 2 carbon molecules leave. So actually, it has to be whatever the pathway cycle is there, it has to continue, and they have to regenerate oxaloacetate. If it is not regenerated, suppose I remove alpha ketoglutarate, I cannot regenerate oxaloacetate, the cycle will not continue. So for continuation of cycle, this has to be continued. So if something is removed from here, there is another pathway which is called as an anaplerotic pathway, will be substituting some of the molecules in this pathway from pyruvate or from some other source, and that's the way the cycle continues. So what we can say is that the citric acid cycle or TCA cycle or hence a Krebs cycle is the one which is amphibolic pathway. It has both catabolic reaction as well as anabolic reaction. Now let us see about the first reaction of this pathway. Here you have oxaloacetate, you have acetyl-CoA, it forms citral-CoA, and then citral-CoA is the intermediate and then forms citrate. Now I have a question. Why this enzyme, the enzyme is citrate synthase, why this enzyme is not cleaving coenzyme from acetyl-CoA when it is cleaving the coenzyme from citral-CoA? Both are more or less similar molecule. Ultimate function is that it is removing coenzyme A from this molecule. So why it is not happening? Any one of you? This is involved in the sequential reaction. So on the surface of citrate synthesis, first oxaloacetate binds. Once oxaloacetate binds, it creates a space where acetyl-CoA can bind. Once these two molecules can bind, then only it makes the changes in the structure of enzyme so that it makes the space where cityl-CoA is going to be accommodated. Once cityl-CoA binds with the enzyme, then it will again make structural change in the enzyme so that the molecule, they come close to each other to cut the coenzyme A portion, and that's the way the reaction occurs. So it is the sequential reaction in which oxaloacetate and cityl-CoA binds, which binding initiates the space for binding of cityl-CoA, that means synthesis of cityl-CoA. Once cityl-CoA is a bind, then only the function of hydrolysis of coenzyme A occurs. Otherwise, coenzyme remains intact, and that's why the coenzyme A from acetyl-CoA is not being removed. So after citrate is being synthesized, it is going to be isomerized to isocitrate. There is a single enzyme which can catalyze both reaction once water is removed and then water is added. You might be wondering why the enzyme is doing this foolish job. Once it removes the water and again adds this water. So actually, by this process, what it does is that it makes a molecule which it can in future be produced for generating high phosphoryl group transfer potential molecule. So actually what happens that it makes an intermediate which is a cis aconitate, and the enzyme responsible for conversion of citrate to isocitrate is an isomerase. So what you are going to do is that, this one, citrate to isocitrate, is done by isomerization of a citrate, which is followed by hydration step. So enzyme catalyzes both step, and the enzyme here is called as aconitase because cis aconitate is an intermediate. So it is named based on the uh, this intermediate molecule. It is an iron sulfur protein, so don't worry about that. It is based on its catalytic reaction, and which has iron in the center. Now, once isocitrate has been synthesized, now these steps are the one which is productive step in terms of the metabolite. What you get here is that isocitrate is subjected to decarboxylation reaction and dehydrogenation reaction. So two steps are there, and that's why it is known as you have a decarboxylation first and then dehydrogenase. Both of them, they are being catalyzed by same enzyme, isocitrate dehydrogenase. So isocitrate dehydrogenase converts isocitrate into alpha ketoglutarate, and here one carbon dioxide is being removed. So you have seen two acetyl CoA has, two carbon has entered in the form of acetyl CoA. One of them is being removed from here as a carbon dioxide. Reaction is illustrated here. One is that NAD is getting reduced and isocitrate is getting oxidized. It forms first oxalosuccinate and then alpha ketoglutarate. Next step, what will happen? Alpha ketoglutarate is converted into succinyl CoA, and here the coenzyme A is added. I will tell you why this coenzyme A addition is important, but again, you are looking at two mechanisms. One is oxidation, another is you are looking at the removal of carbon dioxide. So this is an oxidative decarboxylation reaction which is happening. Next step, once you make this succinyl coenzyme, is very important. This is the only step where at the substrate level you are generating energy. So succinyl CoA is an energy rich molecule. It has delta G, which is minus 33 kilojoule or minus 8 kilocalorie per mole, which is equal to more or less slightly higher than the ATP hydrolysis. So what it will do is that it will transfer its phosphoryl group or its energy so what will happen? It will trap the energy and it will phosphorylate GDP into GTP. So actually this reaction occurs, and this reaction is considered equivalent to the synthesis of ATP because it can, in presence of GTP, if there is ADP in the cell, they can be phosphorylated. So this reaction is responsible for synthesis of ATP. So this step where succinyl CoA is converted into succinate and coenzyme A, which was earlier utilized, is being released, is important step where one molecule of GTP is being synthesized, which of course leads to the synthesis of ATP at later stage. So it is always considered in terms of producing one molecule of ATP. Now, next reaction, first it involves conversion of succinate to fumarate. You will learn that FAD is converted into FADH2, and this is a slightly high energy conserving molecule. So what will happen? It's succinate is oxidized to fumarate by succinate dehydrogenase. Enzyme here is succinate dehydrogenase. It will form the fumarate. The hydrogen acceptor in FAD is FAD rather than NAD plus. Why it is so? That it is used in the other three oxidation. So NAD plus you have seen in other reaction, oxidation reaction, but here in this step FAD is involved. Why? Because this FAD remains covalently linked with enzyme, and then the energy which is being formed at this step, or the energy conserving process of FADH2 is less, because later on in oxidative pathway you will be learning FADH2 is the one which can produce only 2 molecules of ATP, whereas NADH can produce 3 molecules of ATP. I'm just talking about the general cycle. Though this number is debatable, it has been considered to be at different levels. So that part we'll discuss when we'll discuss about the oxidative phosphorylation. So now what you have learned is that succinate, I'm talking about EFAD, that means FAD remains attached to the enzyme, converts succinate into fumarate, and fumarate by removal of one water molecule forms malate, and this malate is again undergoing the dehydrogenation reaction, and one more molecule of NADH is formed, and then ultimately you get oxaloacetate. So now oxaloacetate regeneration is important, because oxaloacetate once formed, again it will bind with acetyl CoA and the reaction will proceed. So this is the last step in the reaction, and that is the regeneration of oxaloacetate. So at the end of the citric acid cycle, what you have learned is that acetyl CoA, one molecule of acetyl CoA. Of course, if I start with glucose, how many molecules of acetyl CoA you will be having? One
Sure,
molecule of
two.
acetyl CoA is formed from, yeah, somebody was saying two, yes. How it is two? Because you are having glucose as 6 carbon molecule. This 6 carbon molecule forms 2 pyruvate, because they are 3 carbon molecule. From those 2 pyruvate molecule, you will be getting 2 acetyl CoA. So when you do the calculation for ATP, you should consider 2 molecule of acetyl CoA formed from 1 molecule of glucose. So the net reaction I was talking about, if I have 1 acetyl CoA, it will also need 3 steps where you have consumed NAD plus, one step where you have consumed FAD, which is converted into FADH2, one step where GDP and PI is combining and making GTP, 2 water molecule is being consumed, and from 2 carbon molecule which was present here, this 2 carbon molecule has been converted. So what has happened? This acetyl CoA is more reduced than the carbon dioxide, and that's why this is an oxidation process. So overall in citric acid cycle, what you did is that 2 carbon molecule which enters as acetyl CoA has been converted into 2 carbon molecule as a carbon dioxide. And ultimately what is your gain is 3 NAD plus is converted into NADH, 1 FAD is converted into FADH2, 1 GDP plus PI has been converted into GTP. So ultimately this is the gain, this is the one which you are looking for. So what you have is that this NADH, FADH2 and GTP is the end result of your TCA cycle, and these molecules, they are important for energy generation. So ultimately what you have seen here is that if I look at these reaction, molecular oxygen is not involved. Why molecular oxygen is involved in the process or continuation of this reaction? Because unless we convert this NADH, this FADH2 into FAD or NAD, this reaction will not continue. So citric acid cycle will block if I do not produce NAD plus or FAD here. So unless we have these molecules, you will not have the continuation of TCA cycle. So actually TCA cycle is considered as aerobic cycle only because the NADH, when it is converted by molecular oxygen into NAD plus, then only this cycle will continue. Otherwise, it will block. But otherwise, you see this reaction, oxygen, molecular oxygen is not being consumed. So that is what is the end result of TCA cycle. And so when molecular oxygen, so that's why TCA cycle will continue only when molecular oxygen is present. So next step will be, I'll be talking about a little bit about the control of TCA cycle, and then we will move on how these NADH and FADH2 is converted into, is its electron goes to molecular oxygen, and that's the way you produce energy. Okay, so any question? So you have pyruvate, you had synthesized acetyl-CoA, it is in the mitochondria, it will be converted into citrate, then the citrate will be converted into isocitrate, isocitrate will be converted into alpha-ketoglutarate. This is a dehydrogenation reaction, decarboxylation dehydrogenation reaction, again dehydrogenation reaction, and then succinyl-CoA formed, it converts into succinate. Succinate is the one, at this step, you get one molecule of GTP. Then further, it will be regenerated by one more step of dehydrogenation. So you have seen that there are three NADH which is being formed, one FADH2 is formed, and one GTP is formed. So that is the overall gain at the end of TCA cycle. Any questions? Okay, if there are no more questions, I'll be signing off. Okay, then. Thank you.
So just a second.
So,
Yeah.
So can you explain like what do we have to do in the group project specifically?
See, which project we are talking about? The assignment which I have given.
Yes, sir.
So in the assignment, what I have given you, I have given you a topic, and this topic is given to a group of 4 persons. Now, first I thought I'll give you a paper which you should read and do it, but then I thought at your stage, because it's the first year, you are not very much comfortable. So I have given you some source from where you can get what is this topic. Though you may have learned about those topics somewhere or the other, but what I have given you, I have given you a clue from where you can get the source. So I have given you a book, which is your textbook for biochemistry. So you can read the book of Stryer. The Stryer explains everything which has been given to you as a topic. Now, from that topic, you understand the basic, and then you look for a paper. So suppose I have given you a topic. On that topic, you look for a paper. Once you look at the paper, you read that paper and write what you have understood from that paper. I do not want a detailed explanation. Just I need maximum 4 to 5 pages, which you can give as your summary, what you have understood from that paper. You give the reference of that paper, and most importantly, you can give a graphical abstract, and this graphical abstract should give the theme that what you have understood. So though most of the paper they are giving graphical abstract, I do not want you to copy it, but what you have understood, that should be presented in the form of graphical abstract. So this is a 10 marks assignment, and I have given you almost more than a week time when you can do it at your leisure. So that is one thing which is required. Second thing is that I have also tried to reduce your burden by bifurcating the course for your quizzes. And these quizzes, of course, there are 2 weeks. So within these 2 weeks, they will be happening. And so your topics have been decided, and 10 marks each will be as a part of your quiz. So that is all about what I have mailed you.
So for the assignment, do we have to write it or can we type it out?
Anything, it is your wish.
Okay.
You
And
can,
will the
yeah, you can
evaluation
submit
be
it.
done? Will the
What is done?
evaluation be done individually or group wise?
See, there is one part which I'll take as a group. So all of you will be writing one para about the subject. So that is based on the group, and the rest part is your individual contribution. So individually, if you have, so first part is writing one para. One para is about the subject that is given in the book. So I would like to understand that, yeah, you have really understood the topic. You know the subject. So that means you are reading the book and the chapter, or at least that portion of the chapter. Second part is the paper. So that is the major part. You will be mostly evaluated in that part. So group I have given you so that there is some consistency of 4 members, and I have randomly selected based on your entry number. I have not selected people from on any there is no bias. It is based on your serial number or entry number, and based on that the topic has been given. So once you write individually, you have to see the paper, give the reference of the paper, and you have to write 4 or 5 pages about that paper which you have read. It should be, as I said, that it should be recent paper. That is good. In some cases you may not find recent paper. I know which are the subject, so there whatever you find you can write that. The most important thing is that it should not be replicated, it should not be duplicated among yourselves. So that should be your individual contribution, because 10 marks
Excuse
are
me.
not, yeah, 10% of your evaluation. Yes, yes, please.
Can two students write on the same paper?
No, you have to, that is where the difference will happen. If you think that the paper is a very big and there are two different parts of that paper, you can take different aspects of that paper, that may be okay, but I will prefer that you should go for a different paper. In each subject you have hundreds papers, so it is not that there is a limitation of paper. You can take different area, different parts of that subject. So each one will be, it's a topic which is a general topic. So you will find several papers. So you can select one paper. So what is important here is that 4 of you should discuss among yourself that who is taking which paper. So it should not be duplicated among yourself.
Answer just last question.
Yeah.
That the one paragraph that you said that has to be done group wise. So do we have to submit it separately or with a
No,
four to five
you can
pages?
submit with yours one. So that is not a problem. Each one will be submitted. That part can be common. That part could be different based on your understanding. I don't mind that. But first part should be your understanding. It's a very small portion, but it will give you some. It is for synthesis of the whole paper. So you can write only one para. One para is nothing about the understanding of a particular topic in biochemistry.
Okay, sir. And
So
so is
mostly it
there
will be
any
evaluated.
specific
Yeah.
word limit?
There is no word limit. I have given you pages
Okay.
for 4 or 5 pages. That is what is the limit. Somebody, if you find something interesting, I do not want garbage. You should not write all those things which are already given in the paper, because I will see the paper. If you have just copied the paper, you will lose all the marks. So don't come and cry if you are just copying the paper. You have to understand the concept and then write. So it is your words. It should not be just copying and pasting from net or from, and that's why I said that you have to read a paper, not something going to some site and copying it. Rather, you have to read a paper, give the reference of paper. From that paper, whatever you have understood, you may have understood everything, you may not have understood whole thing. So whatever you have understood, you can write, and based on that, it will be evaluated. Okay.
Okay, sir. Got it.
Is it clear?
Yeah.
So what will be the format for the quiz?
What do you mean by format?
So
Format is,
format as in will we have MCQ type or we'll have it subjective type?
no, no. MCQ doesn't help because everybody does cheating. That is known. So it will be written part. And maybe I will, today itself, I will write a note to all of you that it is very short answer type because you don't have much time. So in a very short time, you have to answer all the questions. And these will be just one-liner, two-liner answer for each question.
Thank you, sir.
Okay, thank you.
Thank you, sir.